So I wanted to present some work that we've done on both analyzing natural systems and also artificial systems, and you'll see what the connection is as we go along. The outline of the talk is really, it's split up in two parts. The first part I'm going to talk about natural systems and really provide an introduction to looking at light harvesting and transport and light harvesting. And then the second part is going to focus on artificial systems for light harvesting, okay? Particularly the ones that we've worked on. So photosynthesis, most of you probably already know this. It's an amazingly complex process, and it takes place over a large range of length scales and time scales. To really understand it, you need to get to grips with the whole process, because everything is interconnected very tightly. Fortunately for us, if we're looking at artificial implementations of photosynthesis, then we really only need to focus on the first portion of this graph here. The rest of this is really the biology of the system, and maybe we can decouple from that. Okay, so I'm going to focus on this half of the graph here, which involves all the processes going from light absorption to electron transport. And so if we zoom in on that area, what we call the initial stages of photosynthesis, it really looks like this. You have a creation of an exciton as a result of the absorption of a photon, and this exciton is funneled through a bunch of pigments to something called the reaction center, which is where you have charge separation, which is basically where the exciton is ripped apart into an electron on whole free carriers, and this provides a battery to fuel the rest of the chemical processes. Okay. So the main player here is the light harvesting complex, which is, I've shown you an example here. This is PS2 from plants. It's a fairly complex structure, and if you look in nature, there's a huge variety of light harvesting structures. This is just a sampling of a few of them. You have some that are very symmetric like this. You have some that are not so symmetric, or at least at first glance, they're not so symmetric. But some things they do have in common are they're all composed of very densely packed pigment molecules. And most of them are also embedded in some protein scaffolds. So the protein is holding the pigments together and providing some kind of structural and energetic tuning. Okay. The other thing that nearly all of them have in common is that they're amazingly efficient. So that's part of the purpose of this workshop, to really understand this. And they're over 95% quantum efficient, meaning that 95% of the photons that are absorbed are eventually carried over to charge separation events. Okay? And so we want to understand how this happens, and how it happens so quickly as well, at picosecond timescales. And if we look at artificial, or at least organic solar cells, we're really struggling to get to these kinds of efficiencies and timescales of charge separation. So we have a few experimentalists in the talk, and so I'm going to leave most of the experimentalist discussion to them. I'm going to focus on some of the theory because that's my expertise. So let's think about how we would model energy transfer in such systems. It begins with a Hamiltonian for the electronic degrees of freedom. What you have a bunch of pigments that are coupled to each other. So you need transition energies for each one of the pigments, and you need some description of the coupling between the pigments. This is usually at the dipole-dipole approximation, but in reality it should be a full electronic coupling, or at least a Coulomb coupling. In addition to the network of chromophores, of course you have the solvent or protein environment around the pigments, and this provides some kind of bath to the electronic dynamics. Okay? So we need to include the electron-phonon interaction and the phonon Hamiltonian as well. And typically what people do is you derive a reduced set of dynamics from this full Hamiltonian. Usually these are called master equations. There are other approaches as well. And I want to talk a little bit about how you do this in these particular systems. So let's examine a simple excitation transfer system where you have excitation being transferred from this state where the first chromophore is excited to the second chromophore being excited. Okay? So in the end, the product state is ground state of first chromophore and excited state of the second chromophore. And what are the processes involved here? We can draw out classic kind of chemistry potential surfaces. So in the Born-Oppenheimer approximation, at least, the The excited state is created by the absorption of a photon from the ground state manifold, and so this is the excited state manifold of the first chromophore. Right? We assume that it's parabolic. So once it's excited, there's a certain relaxation time over which it relaxes into the ground state vibrational manifold of this first chromophore. Right? And then, or maybe this is a competing process, you have a transfer of this excitation into the second chromophore, and then again another relaxation process into the ground state manifold of the second chromophore. Right? And so what are the energy and time scales involved here? Firstly, there's of course the relaxation time. Well, actually, let's begin with the pigment coupling. So that's the coupling between the two pigments. There's the coupling of the pigment to the protein bath. So that's this reorganization energy. That's a net change in energy as a result of the protein relaxing around the new excited state. There's a relaxation time scale for this. We convert that into an energy scale. There's an energy difference, pigment energy difference. That's typically measured as the difference in ground state energies of this excited state manifold and this excited state manifold. And then there's, of course, temperature. This is all happening at some ambient temperature, right? So there are all these energy scales to keep in mind when you're modeling such a thing. And typically, theories that tries to model this kind of energy transfer use some approximation in these time scales, because there are a bunch of energy scales. There are a bunch of energy scales, and typically you have to make some approximation that one of these is small or one of these is large compared to the other. So I've just put down a variety of theories that people use and approximations that go into each one. The tricky thing about photosynthetic systems is that really none of these approximations is valid. Except for maybe the last one where the fluctuations are Gaussian distributed. But as far as energy scales go, everything is on top of each other, right? So usual approximations, you have to be a little careful with them, because you have to be careful about what you're throwing out when you make one of these approximations when modeling the system. And the thing I want to stress here is that really capturing both the coherent and the incoherent dynamics is critical for these systems if you want to understand the transport. Even if you use one of these approximations, it has to be such that you're capturing both the coherent and incoherent dynamics, right? So what does transport actually look like in this regime where all these energy scales are on top of each other? It certainly doesn't look like band transport from what you maybe expect from solid state theory. It also doesn't look like this kind of cartoon picture of the conventional model, a textbook model of photosynthetic light harvesting, where a photon hits a pigment and then the excitation energy just hops between pigments and eventually gets to the reaction center. So here's a cartoon of what that would look like. You know, you'd have a particle just moving along in an energy well in a space. So that's not correct. The correct picture is firstly where the electronic excited states are delocalized. So they're not point-like quasi-particles. They're actually delocalized in some energy and space. So here is an example of these excitons, these electronic excited states in this simple system of the FMO complex, where the numberings are the sites, or the chromophores, and the excitons, these blobs, are meant to indicate the amount of delocalization on them. So the transport, what would it look like? Well, this is again a cartoon picture where the wave packet is now moving around this energy landscape, but it has more coherent motion. Right? So this is not meant to be completely accurate. Of course, the wave packet is actually also shrinking and moving around and distorting. Unfortunately, my PowerPoint skills weren't up to representing that. But that's a cartoon. And another way that you might want to think about it is, which might be more familiar to quantum transport folks, is to really look at the displacement of the excitation during the energy transfer, right? So what I've shown here is this FMO complex. It's not so important about the details of this, but maybe we'll hear more about it in subsequent talks. But for now, I just want to point out that there's a source of excitations at the top, and there's a sink at the bottom, right? And this thing really functions as a wire to take these excitations from source to sink. And if you look at injecting excitations at site 6, and you quantify the amount of the net displacement of this excitation as a function of time, it looks like this: that it first moves as if it was ballistic, and ballistic diffusive. I've written an analogy to bulk transport, but you should really keep in mind this is a very small system. So these are maybe ballistic with inverted commas.
This is numerical, no?
This is numerical, yes. Yeah, it's not experimental. Right. So there's an initial stage of what's like ballistic transport where the displacement goes as t squared, but then it quickly localizes, right? It plateaus here and almost goes flat. And then that's followed by another regime of transport, which is more like diffusive transport, which is linear in t. And then finally saturates because it's a finite dimensional system and it can't go anywhere else. Actually, that's an important point, that it does saturate, because it means that there is a net, in addition to the sink, there's a net energy gradient here, which is making the excitation flow downhill. Right? So the mechanisms here, like I said, this localization is really due to something akin to Anderson localization, that because of the difference in energy between pigments, it initially starts spreading out ballistically, but it quickly gets trapped in a certain region. And then to get over this localization, you need this new mechanism to kick in, the dephasing, and this is provided by the protein environment. So this is why it's important to capture both of these, because if you threw away the protein environment, you would only capture this regime, and if you were in the limit of very strong forcing by the protein environment, you would only capture this limit, right? And really, to capture the full transport, you need both. Okay, so this kind of complicated transport of this mix of incoherent and coherent is exactly what was probed by these experiments that were done in 2007 by Greg Engel and Graham Fleming's group, and also followed up by many, many experiments since then. And we'll probably hear, I think, more about these today and maybe tomorrow. What I'd just like to point out here is that these experiments probing this incoherent or partially coherent regime of transfer has been performed on many, many photosynthetic systems, and many of them seem to display this. So this is not a unique thing to one or two photosynthetic systems. It seems to be a fairly common feature, right? Okay, and really enticed by that experimental result, a lot of people have performed many detailed calculations on transport in these systems, theoretical calculations, and ascertain that this regime is actually, there's something special about this. This is actually optimal for light harvesting. And what I mean by this is that if you move any of these parameters too far away from overlapping with the others, you would get slower transport across photosynthetic systems. And so I think this is nicely summarized by this early paper from Alain and Spiroguzuk's group, where they showed, okay, so this is just looking at one of these parameters. So it's this ratio lambda on J, so the coupling to the protein environment divided by the coherent coupling between the pigments. That's on the x-axis here. And on the y-axis is efficiency. Let's just look at the blue curve, which is efficiency. And you see that at the very small coupling, you have some efficiency, but not unit efficiency. And very large coupling to the environment, you have very small efficiency, right? And somewhere in the middle, though, it goes through a maximum, right? And this maximum is about where these two are comparable. These two timescales are comparable. So again, this is just underscoring the point that the balance between the coherence dynamics and the environmental coupling is important. Okay, so let's see. Yeah. So how do the next thing I want to move on to is how do photosynthetic systems really maintain this balance? Typically, in condensed phase chemical systems, you expect let's just look at one of these parameters, right? J on lambda. This coherent coupling divided by the incoherent coupling. You can perform the same kind of analysis for any of these other parameters as well, but this seems to be the most intuitive. So, and typically for chemical systems, this is much smaller than one, meaning that the influence of the environment is much more dominant over the coherent coupling. And so, you know, you might even take this as a definition for what separates physics from chemistry. But for photosynthetic systems, in fact, this ratio is kept about 1 by two factors. First is that the interpigment coupling, the coherent coupling between chromophores, is increased by a dense packing of pigments, right? So you see very small separation between these pigments, and that increases the couplings between them. The other factor is that the solvent or the protein coupling is reduced by a number of things, predominantly the fact that these things are embedded within membranes, right? So because they're embedded within membranes, they're less exposed to solvent, and so if you just compare the typical reorganization energy, which is a measure of the net coupling to the environment that a pigment has, if you look at typical systems of pigments in solution, They range from hundreds to thousands of wave numbers, whereas for this example system that I showed you, this FMO complex, it's 35 wave numbers or thereabouts. So there's a difference in this coupling as a result of the particular structure around the pigment protein complex, right? And a large part is played by the membrane here. So the point I want to underscore is that the structure is critical here. The structure is what gives rise to this particular balance of energy scales, and this balance of energy scales is what gives rise to this balance of coherent and incoherent transport, which is what gives rise to the efficiency of transport. Okay, so that really moves me to the second part of the talk, which is analyzing the structure and really asking, can we mimic this structure in artificial systems? Okay, and this is really this line of thought was born about by this comparison here. So on the left here, I've shown a photosynthetic system, a natural photosynthetic system. It's called LH2 in purple bacteria. There's actually LH2 and LH1 here, and it's a bacteria. And you see an amazing amount of symmetry and structure in these systems. Now, I've taken an extreme case in natural systems where there's an extreme amount of symmetry and structure. But in most systems you see some kind of hierarchical structure, some kind of ordered and spatial structure as well, and energetic structure. So there's a gradient usually in these systems, energetic gradient. And contrast this with kind of the state-of-the-art organic solar cells, right, where you have a blend of polymers and acceptors. Usually this is a common blend of polymers and also buckyball kind of fullerene rings. And granted, there is some control over the morphology and the relative magnitudes of whether you have acceptors or donors, but there's no fine scale structure here, right? There's no energetic gradient, there's no kind of spatial structure. So the line of thought is really, can we mimic the structure that we see in natural systems and carry them over to artificial systems, and then hopefully also mimic their light harvesting properties as a result, right? Of course, the first step that you need to do this is to control the assembly of pigments. So this is a very nice system because it really doesn't involve that much fine-scale tuning of the nanoscale structure. Whereas if you wanted to make something like this, of course, you would need a lot of control of the nanoscale structure. And to do this, we actually turn to another one of nature's marvels, which is or protein self-assembly. And so a lot of proteins in nature self-assemble. I've shown you an example here. This is tobacco mosaic virus, which afflicts tobacco plants, but doesn't afflict humans, thankfully. And the essence of this protein is this monomer here. It self-assembles into these capsids. These capsids are basically containers for the RNA of the protein, right, of the virus. So the RNA of the virus is embedded within this viral capsid, which is self-assembled from these protein monomers. And depending on the conditions that you do the self-assembly, and so this is pH and ionic strength of the solution, you can get various structures. You can get these discs. You can get helical structures of the protein. You can get stacked discs as well. So you have some degree of control of the actual protein self-assembly, or the structure that's self-assembled here. And the way we want to use it to template assembly of light harvesting complexes is, we know that these monomers assemble into these discs. So if you mutate the protein monomer and attach a cysteine residue at a site, you can use that as a link, or you can use a linker to attach a chromophore to this site, right? So you mutate the protein, then you attach a linker to it, and then you let it self-assemble. And the protein self-assembled and provides a scaffold for the chromophores. So you have a nice rigid array of chromophores that are built upon this protein. The nice thing about this structure is that there's a lot of tunability in this, right? So you have control over what chromophores you attach, so you can control the spectral properties of the chromophores you attach. You're not stuck to, say, chlorophylls as in the natural systems or carotenoids. I mean, natural systems use a few chromophores, but it's a limited set. You can really go, you can span the visible range and somewhat into the IR and UV as well by choosing your chromophores. You can control the distances between the chromophores. And so the attachment site is somewhat variable. You can perform the cysteine modification at a bunch of sites along the protein monomer, and that controls the density of chromophores. And so this is meant to be a picture of, if you attached at the 123 site, you get this ring of chromophores. If you attach, say, at the 103 site, you get this ring of chromophores. Right? So, and you can change the interpigment separation by doing this. And we're hoping that this is a nice handle on this coherent versus decoherent transport, right? Because as you increase the density of pigments, you expect the coherent coupling to dominate. And if you decrease the density, you expect the incoherent coupling to dominate.
So you already built this kind of structure, or that's
So the experimentalists that we collaborate with have. So this is...
Succeeded.
Yes, yeah. Yeah, so these aren't experimental pictures. They don't have, so these pictures are based on crystal structure of the TMV self-assembling, but unfortunately they don't have crystal structure of the self-assembled structures when you have the chromophores attached to them. Right. So these are kind of just artist renderings or pictures, but and that's part of the research. I'll mention the part of the research is really trying to understand what the chromophore assembly is in these systems. Yeah, the last point is that this is mass producible. This is not a very finicky system. You can produce these systems, you know, in gram quantities in large vats, basically. The other point to note is that, so I mentioned that we want to use this as a potential light harvesting device. So is there a route to really constructing a device out of this? And this hasn't been done in experiment yet, but there is a route to doing this. And really the thing to do is put these protein templated assemblies onto a charge separation interface, and you can work out the chemistry of a electron transfer scheme. So you just have to attach either the same chromophores or different chromophores to the point where you have contact with the surface to promote the electron transfer onto the surface. And then you just put electrodes onto that surface and pull the charge away. So this is, again, as I said, this hasn't been done. This is a proposal right now. But this is the eventual kind of device that we're thinking about that could be built upon these protein self-assembled systems. Right now, they're all in just solution. So the work that we're doing is a lot of it is just microscopic modeling of this system, really trying to understand as you attach different chromophores onto it, what are the relevant electronic excited states, what are the bright states, what are the dark states. So there's a lot of electronic structure on a few pigments or many pigments. And then there's molecular mechanics and molecular mechanics calculations of assembled structures to understand whether you really have this ideal picture of the chromophores all aligned in exactly the same way, or you actually have some disorder in the system. And what we find is actually there is some disorder. So, you know, they don't all sit like this, but if you actually minimize the potential energy of these systems, it actually looks a little disordered like this. So this has led to more work with experimentalists saying, right, if you use different linkers, can you reduce the amount of disorder and make it more symmetric? And really, how do you tune the assembly here? But I don't want to. Oh, and of course, there's a closed loop here between experiment and theory, and we usually confirm that we've got a reasonable model by fitting to experimental spectra that they give. Right now, we're only fitting to linear spectra, but hopefully, that will we will also be fitting to nonlinear spectra soon. But I don't particularly want to focus on that microscopic modeling for the rest of this talk. I'm happy to talk about it at the end or with people afterwards, but the other portion of the work is really concerned with a top-down approach, which is asking, can we model large-scale structures like this, large TMV templated structures, and ask about what the light harvesting performance is going to be for them? Right? So, you know, when you study small systems, you get an idea for what the transport timescales are like, what the relevant physics are. But if you really want to understand the light harvesting structure of a fully integrated system like this, you need to model the large system, right? And this is also an opportunity to ask some of these kind of structure function questions, which is, you know, what structural features really determine the quantum behavior? What kind of densities do you need? What kind of couplings do you need? Or what tuning or what tunability do you need of the protein in order to have really quantum coherent behavior or transport in this regime of incoherent and coherent? And can we enhance light harvesting process? Can we enhance the light harvesting process by suitably designing the system? We've said that there should be coherent transport, but can we really confirm that? And so an example of why you'd want to do some of this large-scale modeling is that when the experimenters come to us and say, well, is it better to build this system or this system. So this is whether you attach a chromophore at 103 or 123. So this is where they're very close to each other, or this is where they are relatively far apart from each other. Which one is better? Which one is going to have the best light harvesting performance? We want to be able to answer that kind of question in a constructive way. And so the first problem that you run into when you do this is, well, how do you quantify best light harvesting performance? Right? If you had a full model of the whole system, all the way down to actually collecting a voltage out of it, it's easy. You want the largest power output, basically, voltage times the current. But at this level where you don't have a full-scale model of the whole process, really what you want to do is quantify best light harvesting performances in multiple metrics. Right? You want fast transport to the charge separation interface. This is akin to these natural systems where you want to collect as much of the photons or the resulting photoexcitations as possible. But you would also want a wide absorption window, right? You want to collect as much of the light that's impinging on your system. You don't want to be just collecting light at 500 nanometers and that's it. So intrinsically, these light harvesting and you can make some analogy with nature too, that intrinsically nature would have to have optimized a number of things, right? It's most likely not just optimizing for transport, because in the end survival is the key thing for all organisms. So to say that this is optimized for transport is only looking at a very small part of the problem. Asking questions about what nature has optimized for is a tricky thing, and I want to stay away from that. So I'm going to focus on artificial systems where we know what we must optimize for, which is both bandwidth of collection and also fast transport to the interface. And the key question is whether you have competing objectives here. Can you do both of these, and can you do both of them well? And so we frame this as a multi-objective optimization. And so this is different from a normal optimization or a single objective optimization in the sense that when you have multiple objectives, one way to do it is just to combine them into a single objective, right? So the key aspect in optimizing a multiple objective problem is what's called a Pareto front. And this is just an illustration of a Pareto front. So what this is that if I plot both my objectives on an axis, so I have two here, one on this axis, one on this axis, and I just throw down all the points that I have, all the feasible solutions that I have, they're all going to have achieved some objective 1 and some objective 2, right? There's a certain set of points that are on so this is a maximization problem. You want to maximize both of them. And ideally you'd like to be way up here, right, where you're maximizing objective 2 and objective 1. But what you find typically in these systems is that you can't go all the way to this corner. There's a front, and this is called a Pareto front, where you can't go anywhere to the right or to the above, both simultaneously, right? So you can't go above this point and to the right of it simultaneously. If you want to go above it, you have to go to the left of it. You have to sacrifice in one of the objectives. Yeah? So the question we're asking really is, what are are there trade-offs in light harvesting? Do we have to play tricks in order to trade off efficiency versus spectral width of absorption, and hypothetically, would natural systems also have had to do that? Right? And so we're going to go back to this structural model that we have, where we have these stacked disks of TOV templated chromophores on a charge separation interface. And this is a minimal model or a reduced model that we're going to work with. And so I assume that I have n disks. So these are these disks. So this is just a model for the chromophores. I'll get to the protein environment in a second. And I have m chromophores per disk. Right, so this leads to some Hamiltonian as a result. And the things that I can tune are the energies of each one of these disks. So each one of these disks, the chromophores in each disk has a distinct energy. But all the pigments on that disk have the same energy, right? Transition energy of the pigments. In addition, all the pigments are attached to the protein in a certain manner. So there are two angles that dictate how the pigment is attached to the protein as a result of the linker group. That dictates a certain orientation of the dipole, the transition dipole of that pigment. And finally, there's a misalignment angle. So the disks that lie on top of each other don't have to be exactly vertically on top. You can have a certain twist to it. You can introduce a helicity into the structure, and so that's another angle that you can have. Now, this is a reduced model and kind of idealized model because the experimentalists can't tune all these parameters all at once currently. Right? So this is very much an idealized calculation to see if you were to have more experimental control, what is the best structure that you could produce? But the idea is really to understand this landscape of light harvesting, whether there are trade-offs. So we're going to throw in more control than we actually have in the laboratory right now to answer this kind of more theoretical question. And of course, both of these structures, TMV 103 and 123, the dense pact and the coarse pact, can be modeled like this. It just changes the length scales of these disks. And the calculations I'm about to show you are for when you have so 10 disks stacked on top of each other and 17 chromophores on a disk. So this is already 170 chromophore calculation, which is already kind of taxing our calculation abilities. Obviously, the real physical systems would have more disks, but we can already get an idea. You already lose some of the small size effects by going to 10 disks. So that's 13 free parameters to optimize over because you have 10 disks, so the energies on each one of these disks, and the 3 angles that define the actual orientation of the dipoles. And so you want to optimize over this fairly large parameter landscape. And okay, that's the structural model. Now I want to go to the dynamical model. How do you actually model the transport in the system? I threw up a whole bunch of models there. There are some that are simpler than others, some that capture some effects and ignore some effects. We have to throw away some of them. We can't use the model that captures all the relevant physics because it takes too long to run. So what we go with is what's called the modified red field theory, which is, if you're familiar with conventional red field theory, it's a slight variant of it. It captures all the reorganization effects well. It doesn't involve the secular approximation for red field theory. And what it results in is a rate equation, so a classical rate equation for the exciton populations. So I should say this has been used reasonably well to capture dynamics in large-scale natural photosynthetic systems. So the PS2 system that I threw up at the very beginning, which is very large, you can use a modified red field theory to capture dynamics across this system. So the benefits are it's efficient. It's a rate equation, right? It has a relatively accurate representation of the bath, the reorganization, and respects fluctuation dissipation theorem. So, which means that you eventually get to the right thermal state. The drawbacks are that it can't model coherences and exciton bases. So, this is a rate equation in the populations of excitons, right? I'm throwing away all dynamics in the coherences of excitons. So the real density matrix contains populations and coherences, and I'm not modeling the coherences. Unfortunately, we have to throw that out because you want something, because you're putting the whole thing in an optimization, you want something that's relatively efficient. And you can check you can check the solutions that you get from these by using a more rigorous theory, but right now we're just using this one. Okay, so this is our first Pareto front. So we throw in that structural model, we throw in that dynamical model, and then we optimize over these 13 parameters, and we want to capture what kind of efficiencies and spectral widths you can achieve. So this is for the TMV 103 system. This is the very dense packing of chromophores. Oops. And these green points are all the solutions that you generate. So this is done by genetic optimization, by the way. So this is there's some randomness to this protocol. This is not a deterministic optimization. But what you see is that you see a very strong Pareto front emerging. So that's these black points. There's certainly a trade-off between these two objectives. You can't go all the way to this corner. As you start increasing efficiency after a while, you have to drop. As you start increasing spectral width, you have to drop in efficiency, right? There's a very steep drop at this point. So there certainly is a competition between these two objectives that you have. And one question is, can you improve on this by going to different systems, right? Can you get a different Pareto front? And so this is the Pareto front for TMV 123. So this is exactly the same plot. I've just removed all these solution points because it clutters up the plot. So the black points are exactly the same black points I had in the last slide. But now the red triangles are the Pareto front of the TMV 123. Okay, so something surprising here is that the increased density of TMV 103, the black points, doesn't really seem to help here. And I should have mentioned at the beginning that there's no disorder in this system. So I'm assuming that you throw in the chromophores and there's absolutely no disorder. This is unrealistic, but I want to show you the difference between when you have disorder and you don't. When you don't have disorder, there really is no benefit to having very dense packing of pigments. It doesn't seem to be that you get larger efficiencies or larger spectral widths. You can always find an arrangement of TMV 123 that achieves the same or almost the same, right? There's a small difference here at this trade-off region where you're trading off efficiency and spectral width, but on the large it's almost the same. But what happens when you throw in disorder? The picture changes a lot. So the blue points are TMV 103, the dense packing, when you have this sort of disorder. Yeah, so I should clarify this, sorry. It's both structural and energetic disorder. So the pigments now no longer have exactly the same orientation that you specify. They're all perturbed by a certain amount, Gaussian perturbations. And the energies are also perturbed by a certain amount. So all the design parameters you put in are all perturbed. And then you run this, so each structure is evaluated over many instances of this disorder to get an average efficiency and average spectral width. And then you run the optimization on that those cost functions. Right. So the first thing is that there's a collapse of the protofront, right? But it doesn't collapse symmetrically. It doesn't go into this corner. The spectral width collapses much more than the efficiency does. In fact, the efficiency doesn't change a huge amount. You can, if you're just concerned with efficiency and you don't care about spectral width, you can choose these points of either one, and you can get almost unit efficiency, which is quite surprising to us. We thought that when you throw in disorder, both the efficiency and the spectral width would collapse. And the spectral width collapses as well, and there's a separation now between the two protofronts. The dense packing of pigments can achieve a greater spectral width on the whole than the coarse packing of pigments, right? So we think that this is, at least in these structures where you have cylindrical arrangement of chromophores, that the dense packing of pigments is really advantageous to overcome effects of disorder and also to achieve larger spectral widths. Right? So if you made this, you wouldn't achieve as large spectral width as you would for the other one. Now, you can also look at these solutions in parameter space. So this was looking at it in objective space, right? So I was plotting the two objectives. But you can also look at what parameters actually form the structures on the protofront. And so there's a lot of data here. I'm just going to show TMV 103 with disorder, right? So that's the dense packing of pigments and with disorder. So it's these blue points. If you look at what the energetic landscape looks like, so from going from left to right on this picture, I'm showing the energetic, and so left to right is going along this protofront, right? So the leftmost, the first column here is for this point on at the very left. The column at the right is the point at the very bottom here. And this is the energies of each one of the disks that you have. So you have 10 disks and 10 energies. The red is the low energy, blue is high energy. And so what you naturally see is that there's an energy funnel, first of all. An energy funnel naturally comes out of this optimization. You want the low energy pigments at the bottom because that's where you have the charge separation event. You want the high energy pigments at the top. There's a shallow energy funnel for the very efficient structures. So the efficiency is high efficiency is over here, and the large spectral width is over here. So there's a shallow energy funnel, meaning that you don't want huge drops in energy. You want a very shallow energy funnel. And there's a much coarser drop. So you have really step-like pigment energies over here. If you look at the angles, okay, so I'm just plotting two structures here. This is the most efficient, and this is the least efficient but largest spectral width. You see that they change dramatically. Right? For the most efficient structures, the dipoles are almost pointing down the cylinder. The transport is not coherent down the cylinder because you have disorder, but this structure is optimal. And the largest spectral width, you essentially have many J aggregates. So each J aggregate is where you have the dipoles pointing head to tail for each one, and so you have a circular arrangement of head to tail dipoles. So each one of these rings is actually functioning as its individual absorbing antenna. Right? And then you have some transport, but you don't really care how efficient that transport is. You rely on basically relaxation for the transport, so it's not that efficient. But each one of these antenna is very has a large dipole. It absorbs a lot, and it's spread out because you have a bunch of transition energies. So you can rationalize each one of these structures, and you can point out exactly why this is efficient and this is larger spectral width. I've kind of only given hand-wavy explanations here, but you'll find more details. There's a paper that's due to come out very soon, working at the end of it right now. But you can rationalize the energetic and structural features of each one of these solution points and figure out, oh, this is efficient because X, and this is has large spectral because X, right? So you can connect the structure back to the objectives that you wanted to optimize in the first place. Okay, and so I won't go through all the rationalizations because there's a lot of data and it's better spelled out in a paper, but I will point out some of the design principles that we've backed away from this study. So by looking at these optimizations, you can figure out, well, okay, at least for these cylindrical structures, and some of these design principles actually generalize to more general structures as well. But you can back out some design principles. So the first is that there definitely is a trade-off between efficiency and spectral width, right? So you can't optimize both at once, at least for cylindrical structures, and we think that's true for other structures too. The optimal structures for efficiency and spectral width change dramatically depending on the amount of disorder. Right, so I showed you the two extremes, when there's no disorder and then there's some disorder. But you can also look at how much disorder, you can tune that amount, and the structures change a lot. So if you go out building a light absorption complex, you really have to be aware of how much disorder you expect in your system, because the design principles are going to change depending on the amount of disorder. As I showed, an energy funnel is desirable in all situations, even in the presence of no disorder, and it's absolutely essential in the presence of disorder. So as soon as you have disorder, an energy funnel becomes the most important thing to achieve any kind of efficiency. And we see in natural systems, nearly all of them have an energy funnel built into them. Some are steeper than others, but nearly all of them do, and that's maybe that's an obvious kind of design principle that you would think you want a directional flow of this energy. The increased density of pigments is not necessary in the absence of disorder. So like I showed you, if you don't have disorder, you can always find some configuration which achieves the same kind of, as long as your pigments aren't really far apart, right? So as long as they're relatively close, you don't need a huge density of pigments. So the density of pigments that we see in natural systems coincides with the amount of disorder that we see, right? So there's some interplay there. Okay, yes, so strong electronic coupling, which is facilitated by this dense packing, is not enough. You need to really control the spatial direction of the strong electronic coupling, which is really what these changes in dipole are doing. I don't have time to kind of go through exactly how the coupling is tuned between these two, but you need to also change the spatial direction of the coupling by changing the orientation of the chromophores. Just throwing in a whole bunch of pigments and making them densely packed is not enough. And then, okay, so this is specific to the cylindrical structure. This tangential alignment angle. So there's two alignment angles. If you say that the disk is like this, and you have a dipole sitting at the end, there's an angle that decides how much you're moving in the tangential plane and how much you're moving in the radial plane. The tangential plane is the only one that matters. The radial angle should really be 0. So this is more for the experimentalists, because when they play around with the linkers that they have, they can say, well, with this linker I expect this much of tunability in the radial angle with this much in the tangential angle. But what we're saying is that you should really only worry about the tangential angle. Okay, so I think that brings me to the end. Maybe one more slide, which is talking about continuing work. This is kind of the dream of what we'd like to do. We'd really like to have an integrated device where we really understand each aspect of this problem, right? So the photoexcitation, the exciton transport, the charge separation, and then charge transfer within the contact. This is maybe a little idealized, but we're working towards this by first understanding the exciton transport, and we're moving on to the charge separation by modeling different surfaces and chromophores on different surfaces at this point. But we have a while to go. But the kind of dream is to really produce a light harvesting device where you understand the microscopic physics of each one of these components. Okay, let me thank all my collaborators. So the theoretical aspects were performed in collaboration with the group of Brigitte Whaley at UC Berkeley, and her graduate student Dong-Yoon Lee, and also postdoc Lauren Greenman, and a summer student Junhoon Lee. The experiments are also performed at UC Berkeley in the group of Matt Francis, by graduate students Dan Finley and Michael Tadeo. So they're really the ones who do the bulk of the TMV modification and experiments. We're funded by DARPA mainly. Thank you.